We are going through another validation case, the number 4. And as in the previous session, we will have first 2 presentations with a different topic. The first one has a more technological approach, while the second one a more scientific approach with data on crustaceans, and in particular on the trophic ecology of crustaceans. We have 2 speakers. I can introduce the first one, and then Elisa can go on with the second part of the session. After these 2 presentations, we will have a general discussion, which will be introduced by Paraskevi Karakle of the Hellenic Center for Marine Research. So we can start with the first speaker. I can introduce Anthony Huguet Vives of the LifeWatch ERIC ICT Core. He is an engineer specialized in front end with a background in computer graphics and animation. So please, the floor is yours. Thanks.
I think that I don't know. Sorry for entering in. I think that if you and Giorgio would prefer to make an exchange, to have Giorgio speaking first, I don't think there is any problem, Salvatrice. No, if
oh no, sorry, I just followed the program.
If the 2 speakers prefer, so Giorgio, I don't know if it is fine for you.
I can do this. Thank you again. It's quite a pleasure to be here, and I will introduce the validation case on invasive crustaceans. Actually 2 species, one from marine and coastal environments, and second one from fresh waters. The first species is Calinectes sabidus, the Atlantic blue crab. It's I mean, to me, it's quite a beautiful organism from a critical point of view, but I started working on it maybe, let's say, 10 years ago, when it first appeared in the waters, in coastal waters of the Salento Peninsula. But as you can guess from the map, well, the native and invaded ranges are shown. We can consider this species almost ubiquitous. From the Western Atlantic Oceans, it appeared in Europe at the start of the past century, and in the Mediterranean Sea in Greece, but then in Italy, in the Adriatic, and let's say in the last 10 years, it has become almost ubiquitous, and most importantly, it started colonizing and appearing also along the eastern coast of the Atlantic, the European Atlantic coast, up to even the English Channel, and in Spain and France, and so on. A key aspect of the species is that it's quite large in size, more than half a kilo. It can reach more than half a kilo, and it's an omnivorous species, extremely aggressive. And today, but I will get back to this later on, with very few information regarding the impact and invaded ranges. Obviously, there are quite a wealth of information from United States and from native ranges, but very few data on the actual ecological impacts, and why not socioeconomical impacts of the species in the Mediterranean in general, in Europe. The second one, so the freshwater species is Procambarus clarki, the swamp cryfish or Louisiana red cryfish. Again, it's maybe I can, yes, keep this out. Sorry. From the southern states of United States, well, it spread throughout the world. Now it's almost ubiquitous in Europe, but also in several other continents, including, why not, Africa, Asia, and United States, up to the Hawaii Islands and so on. It was deliberately, voluntarily introduced in Europe in 1973 in Spain and appeared in Italy in 1989. This is quite a recent map from a recent paper on the actual, let's say, steps and the temporal evolution of the invasion at a global scale. At the basis of this validation case, there are several, a rationale and several assumptions. The rationale that moved me from the very start was that as long as today we have quite a wealth of information regarding occurrences of these two species, far less information are available on impacts in non-native areas. This is particularly true for the blue crab. More information are available for the red cryfish, but again, if we compare the actual ranges of the two species in invaded areas with the information available on impacts, we have to admit that there is an enormous disproportion between the two sources of information. The assumption, the basic assumption, I know that this is not completely true. It's an assumption that it implies a lot of limitations, but to me, as a trophic ecologist, ecological impacts of invaded species should be related with feeding habits. Obviously, I'm talking about animal species, and ultimately with the trophic position occupied by the species itself and the invaded network. Second assumption is that feeding habits and in general the trophic niche of invasive species, but of any species, vary in space and may be affected by local factors, oceanographic or climatic, but also by large-scale, regional scale abiotic factors. The slide is concluded by two keywords. The first is functional biogeography. Functional trophic position, and in general, the trophic niche of a species, it's a functional trait of the species itself. And in the past 15 years, 20 years, more maybe, the biogeography of species has been, and in general, macroecological issues have been addressed in detail using new approaches, advanced approaches and methodologies. Functional biogeography, it's a field, let's say, it's a subfield that is gaining importance, has gained importance only in the past few years. And in order to have, let's say, the necessary data to carry out an analysis, a functional analysis in biogeographical terms, stable isotope ecology and stable isotopes, the analysis of the isotopic signature of carbon, nitrogen, sulfur, but in particular carbon and nitrogen, could be extremely helpful. I don't want to get into the details of the methodology, but in practical terms, it imposes a standardization of the procedures, a standardization that must be applied by any laboratory performing the analysis itself. So no matter if you are in Europe or in the United States, in Japan, or whatever else, the data that you obtain, the isotopic data that you obtain, can be easily compared with those that you may gain, that you may collect from the literature. And this is how the data sets for Calinectus and Progabarus have been built. A scrutiny, an in-depth scrutiny of the published data on focusing on both species, specifically for Calinectus sabidus. The data that were extracted were both at the scale of population, so they are means and standard deviations for the isotopic signatures, and together with data from invaded ranges that have been collected at the individual scale. And this was possible simply because most of the data from invaded ranges have been collected by me and by Professor Vizzini in the past years. And so it was possible to get into the, let's say, the raw data. By the way, this opens to the possibility of analyzing not only the trophic position of the species itself, but also the characteristics of the isotopic niche. But this is something that now we can leave for another, for later on for the discussion if you want. Together with the data on the isotopic signatures of Progambarus and Calinectus were also collected the values, the isotopic values for putative prey species, both animal and vegetal. Applying an extremely simple relationship, is it possible to obtain the trophic position by simply comparing the signature of the predator against that of the prey? And most importantly, this estimation is completely independent on location, independent of any other factor that may be considered confounding. Last point, for Progambarus clarky, we only have in the data set aggregated data. Again, we had also, I had individual scale data, but only for 2 sites in Italy, Lake Bolsena and Lake Trasimeno. Maybe in the future, more and more data at individual scales can be added, but I will get back to this later on. Well, for Calinectus sabidus, this is at the actual distributions of the data. 99, we have information for 91 sites located in, most of them located in native ranges. And for the Mediterranean Sea, we have 12 locations. Specifically, the 12 populations are primarily from Italy, but also from Spain, Greece, and Croatia. Hopefully, this list will get longer in the next few months, because I will collect data from Crete. I'm expecting data from Sardinia and from France. So it's in evolution. These are the locations for Procambarus clarki, 51 sites, all located in invaded ranges. This is quite odd, actually. The scrutiny of the literature did not produce any information on population from Louisiana or from southern states of the United States. Again, also in this case, I'm waiting for data from colleagues from the Louisiana State University. But let's see. So there is quite a discrepancy into two data sets, mostly focused, I mean, almost completely focused on invaded ranges for Procambarus, and including native and invaded ranges for Calinectus. Regarding the progress, okay, for Calinectus sabidus, the dataset should be considered complete. For Procambarus clarki, it will be with the help of Christina Di Muri and Jovic, and it will be completed soon, hopefully within May. Behind the, I mean, I'm making an effort to complete both datasets, but at the same time, because there is a Penelope canvas effect that can always impose to reopen the dataset and include new data. Fortunately or unfortunately, Calinectus sabidus is raising quite a lot of interest in Europe. So there are several groups going on gathering information also on isotopic data from France and Spain. So I want to complete the both data sets, but on the same at the same time, the dataset should be considered also opened and to be updated continuously. Okay. The workflow that should be implemented, it's quite simple. I mean, I leave the details, the technical details, to Anthony that will follow me, but in practice, in brief, 5 main steps involving the sum of life watch services to be used in step 1 to check the taxonomy, obviously not of the 2 predators of the 2 focus species, but of the prey and of the prey species, both vegetal and animal. After the validation of the data sets, in a second step, the trophic positions will be calculated, and an intermediate data set, including only the coordinates and the trophic position estimations, will be checked in terms of coordinates. So there will be a geographical validation of the dataset itself in step 3. In the fourth step, the data, the dataset, the intermediate dataset will be translated into in a shapefile. So in spatial terms, and it will be how can I say, compared, superimposed to oceanographic or climatic layers. So obviously for Calinectes sabidus, oceanographic layers from, I don't know, Marsback or BioOracle or whatever else should be used, while for Progambarus, climatic layers from World Climb or else are expected to be used. And the last step, a modeling procedure will be implemented. In origin, I planned to use, different alternative approach. Starting with the generalized additive mixed models and so on. But there are several alternatives. And at the very end, the final output will be a model, actually, a predictive model of the trophic positions of the two species, modeled and predicted according to climate and to oceanographic, climatic and oceanographic predictors. Only to give you, by the way, I've already started with some collaborators from University of Palermo, so Professor Vizzini and PhD student Dr. Bardelli, to analyze the data, obviously on Calinectes. Obviously, I don't want to burn the data. I'm expected that LifeWatch will let's say, implement the workflow and will actually produce the models. But a first analysis, a preliminary analysis of the data set already produced some interesting results. By the way, some interesting results, but necessarily to be, let's say, inspected and discussed in details, because the relationship that you observe, the negative relationship between the absolute latitude and trophic position, contradicts a lot of theoretical predictions regarding the variation of the trophic position of omnivores according to latitude. But we can get into this, enter into this later on if you want. To conclude, what has been implemented on Calinectes and Procambrus can be extended to any other species. No matter if you are considering marine or freshwater or even terrestrial species. Obviously, the crucial point, the pivotal point, it's the availability of literature data, or why not, the availability of colleagues and collaborators interested in gathering more data and to let's say enlarge to implement the data set. And most importantly to me, obviously, this opens to a general conceptual framework regarding the biogeography of omnivores, not necessarily non-indigenous or invasive species. As regard with climate changes, this could be, according to me, in my opinion, quite an original and novel field to be explored. And in this respect, the LifeWatch infrastructure, could be extremely, let's say helpful. Simply considering the possibility of storing, including in the data to be stored by LifeWatch, also specifically isotopic data. Again, stable isotopes are extremely easy to be, let's say, compared, are quite in prospect to be, are quite fruitful, provide a fruitful field and a fruitful basis to implement large data set and to be analyzed and to be compared with other information, other data, conventional, in terms of abundance, occurrence, and so on. Thank you so much. And again, special thanks to Roberta Bardelli and Cristina Rimuri, Iva Jovic, and Anthony. Thank you so much.
Thanks a lot, Giorgio. Sorry, but we did not introduce you at the end. So Giorgio is a researcher at University of Salento with expertise, as you see, on the trophic ecology of invasive species, with a special focus on crustaceans.
Salvatrice already done, but let me introduce Anthony Huguet is the person that is going to do the second part of the session and more focus in the technological side of what Giorgio already explained, that was more in data. So I give the floor to Tony, and I hope that you enjoy this second part as we have enjoyed the first one. Okay, Tony, you can go.
Hello, back again, and thank you, Giorgio, for explaining the scientific or conceptual part of this workflow and the value that it has. I'm going to talk a bit, going back to the workflow that we demonstrated, or to the scientific use case. I'm going to go and talk about what is built now as a beta. So what you can see here is the conceptual diagram of all the steps and what should be happening. But if you go today to the beta platform of LifeWatch, what you would see is more diagram like on the right one. If you remember from the talk of IJI, my colleague, he talked about wrappers and about services. As you can see here in this diagram, every box would be one of these services that internally calls a wrapper, and is, let's say, the atom of what our system can run. We tried to map it as much as possible to the scientific model, and we have some documentation and some user interface guides to help always the scientific or the user that is behind the screen to understand what is happening. One of the boxes that we see here that are not in the scientific scheme are the importing file. This part might or might not look relevant, but is most of the times the starting point for a workflow. Let's say your custom data, what you want to run this algorithm on. And currently we import it from our file system, but we want to allow the users also to import the data from any given URL. So let's take a look to let's say the state of all these wrappers that Giorgio described a bit. I have to say that most of them are quite ready and in production. This workflow is quite mature, at least the first stage that we wanted to approach. For instance, you can see here in the IT component is the trophy position analyzer, which gets the data and gets the trophy position of the invader. The next one would be the Geographic Checker. So gets this trophic position information that was given to it, and let's say creates the coordinate vectors and checks the geographic information. Next, we can have we can see the spatial viewer and the coordinates verifier. Basically, there's the main mission of this service is to verify the coordinates and match the location. This component has, besides the data part, has a prototype that is a UI where the scientists can see, let's say, the samples on a map and mark them as valid or not. I hope you can still see my screen if I switch to the browser. But yeah, the main idea here is that the scientist would explore the data and would mark which samples are valid or not, and continue the execution of the workflow. Right now, this is a beta, is a demonstration of the functionality, but is not working yet with our workflow, since we are missing the capability of stopping in a certain wrapper or a certain step of the workflow and resuming. Once we developed this part, then the integration with this visual validation and other work in progress for other workflows will be integrated. The next step would be the environmental data extractor, which would get from the validated trophy position and samples and the data provided in the bioclimatical prediction file, and is going to create one of almost the last outputs that is used by the modeler to create our prediction map. Let's take a look. If you were about to run one of these workflows, your first question would be, okay, how can I upload my data to the system so I can run this workflow with my own information? If you log into our beta program, one of the first screens that you can see is the personal space. This is really similar to the folder structure of any computer, and allows you to organize the data that you want to run any of the workflows with. In the future, you will be able to share this information with other researchers, and yeah, also to share the design of different workflows. As you can see, you can upload the data and informs you about the progress of this upload. In this case, we are uploading images for another workflow that is called Ailantus, that another colleague will explain to you. And we give this feedback because the information that you upload can be really huge. If I don't remember wrong, these are about 2 GB of satellite images. Once your data is there, the next question is, okay, how do I run a workflow? To do so, and I'm going to explain the case of the crustacean use case, but this is extrapolable to any of the workflows that will be demonstrated. You would go on the left panel and search for the run a new workflow. And the first part that you would see is actually the diagram of the workflow. And if you hover over the eye of information, you get a bit of a description of what is this service doing and what is going to happen with your data. If we would click next, the system asks you for a description of the workflow. This is a term or some words that help you remember what were you doing. In this case, we name it Mediterranean May campaign, but can be experiment Monday morning or research group C or whatever that helps you identifying this specific workflow. The next, what we would do is upload or select the data that we want to run this workflow with. In this case, for construction, we should introduce the biological data on CN stable isotopes and also the trophic position data. Once we go to the next step, the workflow will be created and will start running in our system. Okay, so the workflow is running now, and probably you are asking, okay, how do I know what's the state of this workflow? Some of the workflows that LifeWatch is working with can take several hours or even days in the future to complete. So how do I track the execution and how do I track the state of the workflow or my experiments that I'm running? Easy, and also for all the workflows, you can go on the left on the dashboard and you will see a list of all the workflows, in this case grouped by the crustacean functional biogeography. So that will be a list of all the workflows that you run in the past. And as you can see here, the first one is the one that we are running now. You here also have the option to revoke a workflow. The use case of doing this is after pressing start, you send all the information and the big computers are busy running your workflow and you remember, I forgot to include this in the data or I did something wrong. Then you could revoke this workflow. So you save the execution time for other people and then you would start over running a new workflow with the new information or correct information that you want to run against. Another useful tool is this one. If we click here in any of the workflows, we go to a tool that is called Workflow Insight. This tells you for the current workflow, what's the state. For instance, we can see that for this workflow, 6 of the 7 services have been completed and that the last one is work in progress. If you hover on the icon, you can get the state of it. And here you can see, for instance, that this is started and is, let's say, the system is busy with it. You can also see the completed ones. And in case of an error, it will also show you where, in which one of the different services, the workflow failed and stopped. If we scroll a bit up, we can see the workflow output files. Each one of the services can have one or several outputs. So inside of a folder in the workflow output files, you can find always for each one of the services a log file which tells you all the information that this service has to offer you. This is useful, especially when the service failed, when there was an error and the workflow stopped. There is where you can find everything that that happened. And also you get a file, one file for each output of the service. If the service has 3 outputs, it's not the case of this workflow, then you would have one file for each one of these ones. As you can see, for each one of the services, you can select the log or the output file and download it. In this case, and for this workflow, that would be the output, which is the prediction map. As last element of this workflow insight component, we have the task list details. This tells you what is the computer busy with and can offer some extra information. Like, for instance, there are some services, like in the ARMS workflow that my colleague IJI demonstrated this morning. that the input parameters are not data, is not a file. It could be, for instance, a geographic position, like a latitude and longitude and a radius, so a range of geographical space where the workflow or this service specifically has to run against. Okay, these parameters would be displayed here in the params. You could see the input, latitude, longitude, and the radius of it. And the last question, or the last piece of UI that I want to show you is, okay, I'm absolutely new to this system. What do I know this workflow does, or how do I learn more about this workflow? There is the last component that I want to show you is the workflow information. It's the information page about the scientific case, and if you click here in the information page, you will be redirected to the LifeWatch Eric dedicated information page for this workflow, and you can learn everything about the input data that is expected, why is this case valuable and important, and let's say the state of the documentation for this specific workflow. But I'm not going to go in detail about this website, since another colleague will do it tomorrow. That's my last slide. Thank you very much.
I can see a first question in the chat. The question is from Alberto Basset. I can synthesize it. Can the crustacean NIS workflows would be generalized for the study of native species, patterns of trophic niche, trophic level, patterns of variations on biogeographical pressure gradients? I think that Anthony can first answer this question, and also Giorgio, if he wants to add something more.
Yeah, I think this is more the area of expertise of Giorgio and more about his part of the talk. I think he already explained that there is a big hope on reusing the algorithms designed on this workflow for other use cases, which, yeah, I think is highly, it has a lot of potential. It's highly useful.
I mean, from the point of view of Anthony, you can give any name to the species, no matter if it is native or invasive, if it's terrestrial or freshwater or marine, and the workflow will flow the same. This is what I briefly introduced at the end of my presentation. What has been implemented in the workflow is something that can be reproduced at any scale, at any ecological scale, in terms of type of species, category, native, invasive, or even for any category from the point of view of, I don't know, commercial species against endangered species or whatever else. The kernel of my point is that now we all witness great efforts made to map species and to predict the distribution of species in space against future climatic scenarios. But this is only one face of the coin. From the point of view of I mean, to get back to invasive species and to alien species, to study and predict the distribution of the species and to have a clear picture of where the species is and where it will be if the temperature will rise by 5 degrees centigrade, it's important. But it's also important to evaluate the impact the ecological impact. I'm getting back to the original definition of ecological impact for invasive species provided by Parker in 1999. That it's a simple product of the per capita effect of one invasive individual on native ones per the distribution and abundance of the invasive species. So as long as this specific information regarding the per capita impact is lacking, you can make quite a lot of efforts to predict the distribution or even the abundance. But you will never have the exact or let's say robust evaluation of the impact, the real impact. That's it. But to get back to the question, yes, it can be extended to, obviously, if we stick to stable isotopes, it can be extended to any typology of species, even communities, or even assemblages. So you can scale up. This was a general idea that obviously has not put forward by myself to use stable isotopes more to get into the detail of species-specific characteristics, but also of community-specific characteristics, of assemblage-specific characteristics. And again, this is a methodology that can be extremely useful in this perspective.
I have a very short question for Anthony about the validation process of the data. How does it work? And also, I saw that in the maps you kept the data not validated. I would like to have more details on this, why you keep this data and what is the process. Thanks.
Yeah, good question. Right now we are running, let's say, in the ideal case. So right now a workflow has a definition, and we have a strict way on how we expect the files to be, which format and columns they will have. In the coming steps, there will be a taxonomic checker and some validators for each file that you want to provide to any of the workflows that will give you more exact information of what went wrong if you are providing a file where the workflow or a specific service is expecting a column or information in a different format that that's how you provide it. Nowadays, what you would see is that the workflow would fail if that's the case. And in the log file of that specific service that failed, you would see a message, but that message can be leading you to suspect about the data or can be absolutely confusing. And this is due to the nature of what we're trying to do of coding. Let's say we want to encapsulate in services also databases or algorithms that are not in our systems, and we are not in control about these messages. So one of the developments of the team of NICOS will be to validate and to give user-friendly error messages when the data is not conforming what we expect. And we will also link you to the page that explains how this data should be and why we do it in this way for this specific service, which I think Lucia Vaira is going to explain more in detail tomorrow. Regarding the filtering process, yes, I did not go really in detail about that, but basically what we could see is a map with all the samples, and some of the samples were green and some of the samples were red. And the green samples are the ones that you consider valid, and the red ones are not valid. In this current stage, what we would like to do is, okay, this is filtering. So the red ones will be taken out of the data set and won't be provided to the next service. But ideally, I think in the future, we should have some interaction for you to check what went wrong. Why is this sample not where you expected it? Is some error in the formation? Did something happen that that the data was right and now is not there in the right format, or did something happening when you were taking the samples, or let's say you will have to diagnose what happened with this information, but we would like you to have tools to correct it and then go on with the workflow without having to repeat all the previous steps. Yeah, that I think are the steps that we want to take. I hope it answers the question.
Yes, absolutely. Thanks. We can see how the NIS species, in a certain sense, are less tolerant to environmental variability, at least in terms of temperature. So probably one of the main points in the successful of invasion is the trophic plasticity of these species. And in this term, I think that more information on the niche of the species is very helpful to understand the invasion success or the potential invasion success of many species. And also I have another question on the potential impact of herbivorous fish in the Mediterranean, because we know that in the Mediterranean native species are mainly omnivores or carnivores. We have only a few herbivores, especially in the western part of the Mediterranean. So I guess what would be the impact of the increasing number of herbivores? Also, because plants are changing in the Mediterranean. We know that the endemic Posidonia is under decline, while other species, such Cimodocea, but also not native species such as Alophila, are spreading. So there is a concomitant change in the primary producers and in the pressure from herbivores. What can be the impact at the basin level of this invasion?
I only want to get back to basic ecology. In practical terms, omnivores have always created quite a lot of problems to food web theory because they simply obtain energy from too many trophic levels. And put down in this simple and trivial way, if you ask me to depict the perfect invader, so a new species with a lot of problems of adaptation to a new environment, new temperatures, new abiotic factors, the possibility of gaining energy from as many energy and matter pools as possible, for me, it's maybe the best badge for invasive species. Omnivory to me, it's linked deeply to invasiveness. So the possibility of for a species to reach the state to establish, reproduce, and to spread in new environments. By the way, in the graph showed by Baraskevi, there were many more omnivores than expected, because you had omnivores between the second and the third trophic level, but also those located between the third and the fourth. And so in intermediate locations, they can be still considered as omnivores because they simply feed on different trophic levels. So to me, it's quite an extremely interesting and stimulating point, omnivory and invasiveness.
There is one last question that Sarah, it was in the chat. So, and it's from Andrea Ramsak. And he thinks, or she thinks, that consumer must be used to a new species, and thinks that if consumers don't know new species, they will not buy them. And even more, seafood literacy is very low according to recent publications. So I suppose that if you want to make any comment regarding this.
The problem with the commercial use of invasive species, it's, I mean, maybe to put down simple, you should consider a fisherman as maybe the best predator for invasive species, as long as the fisherman himself or herself decides that the species has a commercial value, is of economic value. And this sooner or later happens. This is what is happening with Calinectus, but it's happening with the Chinese carp in the United States. It's happening with Carcinus manus in the United States. They're starting. I mean, they are entering the market, and as long as the species is recognized in economic terms, at this point you can consider the human exploitation as a way to control the species. By the way, controlling marine species, it's rather impossible. I mean, at least for animals. So the best way, and we have a lot of experience, I mean, of examples. Pauli in 1980 simply showed us that the best way to bring a species, a marine species, to extinction is to fish it. So human exploitation could be considered, and this is my view, but it's something that is becoming, I mean, in 1917 was something a little bit terroristic to say, also because for the European community, invasive species were invasive species to be eradicated or controlled or anything else. But by the way, the human control, obviously it must be monitored, because another classical example is people translocated Procambarus clarki in Italy from one place to the other simply because they have discovered the species itself as a good for cooking linguine or something else. But this is something that should be controlled, and there is quite a vast literature. I mean, from Simberloff on eating invasive species, how to eat invasive species, and how to use humans to control invasive species, simply letting them entering the market under certain controls, obviously. I mean, humans do not contribute to the expansion of to the spread of the species voluntarily, obviously. On the other hand,
I don't want to stop the discussion, but we are a bit out of time.
Okay.
And we are going to go. The topic was really interesting, and also of perspective of actually sustainable use of invasive alien species.
Let just me thank Alberto for inviting me to chair this session, and the speakers for the excellent presentation. There was a very interesting discussion, and probably we have often this message at the end of a session: we need more information. there is a lot of work to do with invasive species, also in terms of social acceptance and the entering in our habits, also in our food habits. So the need to let the society know more about these species. So thanks again. I really enjoyed, and Elisa, if you want to add something, please.
I just want to say the same, that you had been really interesting for me, and even more that I'm not coming from the scientific side, and I have learned even a lot, and I will check more deep and trying to understand everything. And thanks, Alberto, to invite me as a rapporteur of the, and of course, I will wait here to help you and to finish the end of the day, and hope to see you soon again.